I'm gonna move this.
The RCS. Sometimes 60% of your run if you.
60% of our astronauts are RCS.
Post QC. That's right, that's right.
All right, I'll start now. And so my project here was predicting reads aligned to the RNA Calibration Strand, RCS. So what is an RNA Calibration Strand? For those of us that don't know it, so basically for every single sequencing experiment, I hope, you put in spiking controls.
Every direct RNA one, not every one, but every direct RNA one.
Okay. And so you put in your spiking controls along with your RNA or DNA sample when you're sequencing. And so the RCS has several uses. I've just listed a couple here. One being normalization. So this would be used as a reference for the sequencing depth that you receive. And also, when you're designing an experiment, it helps you figure out the amount of RNA necessary to receive a desired signal. Carly and I were talking about that the other day. So that's really important for a lot of these methods and experimental design. And so the second thing also is it gives you an assessment of the technical variation due to library prep. So there's other things as well, but we're not going to get into those. So in our experiment, the RNA calibration strand is from yeast. It's an mRNA from yeast. And specifically, it comes from this NLase2 gene here that you can see here, on chromosome 8 here. So our data includes these spiking controls, and then let's say our sample here is going to be the hepatitis C virus, which was one of the samples that we looked at in class, the HCV 002 and 004. We're just using the HCV 002 in our data here. And so as one would process this, how it would look like in an experiment is it comes out after sequencing, and you will have signals aligned to the RCS, which is this yeast mRNA, and signals that are not aligned to the RCS, which is this hepatitis C virus. So our goal here, using these two data samples types, is to input the signals output of the aligned reads into a neural network and create a classifier that will output whether a new signal introduced into the neural net is part of the spiking control reads or part of the HCV reads. So some problems that we came across in the start here are that nanopore signals can be huge. So like 500,000 numbers for one signal. So if we want to take a lot of samples, we have to have a ton of storage. And if we want to do anything with those samples, we have to have a ton of memory. So Google Colab might not be the best place to do this. So we had to take it on our own computer, or my own computer, but we'll talk about that a little bit more later. And another problem is that if you take a combination of nanopore signals that some are large and some are small, this leads to many missing values, which is not allowed in any machine learning model that we used in PyTorch here. And so one might solve this problem by imputing the data that you can see on the left here. So basically, it's taking the shorter reads and computing values such that the read is the same size as the longest read in your data for all reads, or you can truncate all of the reads such that they are equal to the same size as the smallest read here that you can see in this example. And so we'll look at both of these scenarios in two different data sets. So another thing to know is there is a 3' bias in nanopore sequencing. So many signals are shorter than longer, which Teo mentioned in one of the lectures here as well, and gave me this cool GIF that I really like watching to completion here. So basically, our data is going to be biased towards the 3' end because these signals are smaller, and like we talked about, a lot of signals is going to take a lot of storage, and it's just not feasible for where we had our signals in Google Colab. And so the data sets that we created here are containing reads with signals between 18,000 and 20,000 values. And so taking these RCS calibrated strands, we had 1321 samples of those, and also 1321 samples of reads aligned to the HCV. And so in this data set, you have a maximum of 10% missing values allowable here. That is 2,000 of 20,000. And so we wanted to impute the missing values here to see how that would work in a model for Nanopore. One of the downsides of this model is that there's not really that many reads, especially if you put in a deep learning model. So that's just something to keep in mind throughout as a limitation. And our second data set here is a truncated model where we took reads between 15 and 30,000 values in total. It's about 9,000 samples per group here. Group, yeah, I'll just say group. And so in this, we have more missing values, and therefore we truncated as a result, such that it's about 15,000 being the longest read, or like number of values for read in this data set. But there's a lot of reads, and so this might be just better in general for deep learning. So how we assess the model is through a process called five-fold cross-validation, which I'll touch on really briefly. Basically, how it works is your data here is split into training and test data, but instead of splitting it one time, you split it 5 different times, and you take 20% of the data as the testing set, and you repeat this for 5 different subsets of your data, so that you can get some calculation of the variance of a metric that you are calculating in your data set. So we calculated a lot of metrics to determine the efficiency. Due to time constraints, we're only going to talk about the AUC, which reflects the model's ability to discriminate between the RCS and the HCV reads. So imputation methods we used for the impute model included the median imputation, which just replaces all the missing values in that read with the median value within that read. We did the mean imputation, which is the same thing as the median, but you just replace it by the mean instead of the median. And then we also tried a k-nearest neighbors approach, which took the 5 most similar signals and replaced missing values based on the 5 most similar value, the average of the 5 most similar values to that read that had missing values there. And so we'll just focus and what we show here is the k-nearest neighbors imputation model here. So some of the architectures we used are 2 hidden layers in the middle here. So you have an input layer, and then your hidden layers and an output layer for this neural network architecture that we used. So we have 2, 3, and then 4 hidden layers that we tried. It's pretty computationally expensive, so it takes a long time to train, and longer as you add more hidden layers because you're adding the number of parameters. The reason we wanted to add parameters or the number of layers in general is because it's thought that as you add the number of parameters, you can learn more sophisticated or more complex patterns in your data. And so this could potentially, increasing the number of hidden layers may potentially lead to a better model performance. So the results here for the impute model, we see that with 2 hidden layers, our AUC value here And you can see in these box plots that the AUC is always the first in each model that we ran. The variation is given. So this is the standard deviation in the error given by the error bars. You can see that we show here just the AUCs on the bottom, and they are decreasing on average across each model as you increase the number of hidden layers. So what this means is that there could be a possible error accumulation due to imputed signals as you introduce or as you add more parameters to the model. This could be due to learning potentially wrong imputed signals here that will make your model like less generalizable in the end. So imputation might not be the best. But for the truncated model, we can see that as you add hidden layers, from 2 to 3 hidden layers, your AUC increases. And then for the 4 hidden layer model, it's pretty similar to the third as well. But there's kind of this like sweet spot of hidden layers at 3 that we see here. I also ran it for 5 and did some other things, and it still stayed like consistent around 0.9785 for 5 hidden layers. But you can see that there's some plateau around 3 hidden layers. One other thing to look at here is that the variance in the metrics here are all increasing as you get to the highest model number of hidden layers in the model. And so this could mean that there's some overfitting as you increase the number of parameters here, leading to more generalizability for the model. So some conclusions are both of the models actually perform pretty well in terms of the AUC, and imputing data may result in a lower performance, especially as you increase that number of parameters and learn something that might not be true signal. A general conclusion is that deep learning models we've shown that they can successfully be applied to these nanopore signals to learn differences between reads. And some future directions, I think, number one is big, is to get more computational power, because we want to add more signals from both groups that we looked at. We can test more complicated model architectures for deep learning, and that kind of ties into more computational power as well. These models will take longer to run probably. We can test on different RNA calibration strands, and we compare with reads with sequence from different animal models as well as humans to see if this holds up for many different species. So with that, thanks to everyone who ran the course. It was really, really interesting, and I learned a lot. And if you have questions, I'm happy to answer.
Well, time for questions. Maybe we'll jump right into the next one for people.
Sure, I can have a question.
Yeah.
So for the imputation, you mentioned that you looked at 3 similar reads to input the signal, or 5 similar reads to input the signal.
Yeah.
Did you look only in the like neighborhood, so like K-mer base, or across the whole read, and which similarity like metric did you use?
Yeah, that's a good question. So if anyone can't hear, the question is, for the KNN imputation model, were the imputations just looking at K-mer based or across the whole read? And so it was across the whole read for the whole KNN, or for the KNN, yeah. And it was only 5 signals. I didn't test other numbers. I didn't really have time, but yeah.
You could iterate through different parameters.
Right, it's a good hyperparameter to test as
Like
well.
a relation based similarity, right? Nothing.
Yes. Yeah.